Hello everyone, welcome to the National Heart and Lung Convenience Store. So my name's Mike and I research the tiny organisms that live inside your lungs. So all of us have bacteria that live in our lungs all the time, and that's completely normal. They're there when we're healthy and they're also there when we're sick. But what happens when we're sick is that they change to different types of bacteria. So we get a shift in which ones are present. And my job is to sample these and to find out about those shifts. What happens when you're healthy and what happens when you're sick. So I'm going to show you how we take a sample and how we get the information about these bacteria, because they're too tiny to see and we can't grow them very well. So, first of all, this is my bacteria. I've got a tin of soup. So what I'm going to use to tell these different bacteria apart is the DNA, is their DNA. It's the instructions within inside the cell that tell that cell what it is. And bacteria have a tough outer shell. So the first thing we're going to have to do is to crack that open. So here we go. So you're going to take a sample, and this is a throat swab. Phil? Yeah. We've got to show it for real, right? How's your gag reflex?
We'll find out.
Here we go. Right. So right at the back of the throat, because the bacteria at the back of your throat are very similar to the bacteria down within your lungs. So that's Phil's sample, all sorted. The other way we can sample, which I'm not going to do to Phil, is the lung brush. So sometimes, if you're quite sick, a doctor will put this down into your lungs, so it gets fed down a tube into your lungs, and then if you look at the end very carefully, there's a little brush there, there's a little bottle brush that they brush back and forth, and then they pull back out, and then we can use that sample to see what's going on down in your lungs as well. So if you remember, I said these bacteria are really tough. So in order to crack them open, I put the sample, which is either the swab or the lung brush, into one of these tubes. So I'm going to hand these out. There's one for you as well. Do you want to pass them round? So they've got lots of little glass beads in them. And there's a big one, which is there to mash up the sample. And then there's lots of very, very fine beads. And when we shake that very fast in this machine, a bead beater, then the beads all smash around and break up the sample. So there's our sample. And here's the bead beater. It's a bit like a blender. And I'm going to set it off. And it really violently shakes the sample around. And that's cracking open this tin. So now what we've got is a big mix, a big soup in there of bits of the tin, all the veggies and the pasta shells, and then the soup itself. What we want is the DNA, which is just the pasta shell. So we've got to get rid of all this other rubbish that's in there. The first way we do that is by centrifuging it. So you can spin it really fast in this little device. So I've got my sample in there again, all cracked open. And then we spin it really fast, if we're lucky. And all the heavy bits go down to the bottom. So I've lost all the tin out of the sample. The next step is that we want to capture the DNA itself. We've got the soup there, we've got some of the veggies left, and we've got the pasta shapes. And this substance is a binding matrix. So it's a very fine sand which will specifically bind to DNA. And I mix the sample in with this. The binding matrix pulls the DNA out, and I can centrifuge it again, and that all goes down to the bottom. So I captured the DNA out of the sample, and I've thrown away the rest of the soup now. And then very finally, our last step is to read the information in the DNA. So I've got this mix of a DNA sample, and I use a DNA sequencer, like this very expensive model here, that reads all the DNA from all the different bacteria in the sample in one go. So that's how I go from Phil's throat swab, or his lung brush, which he was too cowardly to do, down to seeing which bacteria are in the lungs. And if I take lots of samples from Phil over time, I can see how they change and what might happen if he gets sick.